Good afternoon, everyone. My name is Luis, and I'm going to present to you my work titled Development of a Standardized Approach for Transfer Learning with Qsar Models. So first, what is transfer learning? Transfer learning is a machine learning technique where instead of training a model with information derived only from the task of interest, as in traditional machine learning, we also use information from a different but related task called the source task. In a Qsar setting, this would be equivalent to using information from a related target in order to improve a Qsar model of a target of interest. So considering this, two obvious questions arise. How are we going to transfer information from the source task to the target task? And which target should we use as source tasks for a particular target task of interest? The first question pertaining to transfer learning algorithms is definitely the one that has been most discussed in the literature. Therefore, in this work, we focus our attention on the other question. So with this in mind, the objectives of this work are creating a model capable of predicting the successfulness of a transfer between two datasets and creating a standardized transfer learning protocol capable of being implemented in an algorithmic manner. So in order to establish a performance baseline, we created standard Qsar models for all targets present in the KBL database that are either single proteins, organisms, cell lines, protein families, protein complexes, or tissues. Subsequently, we randomly split the KBL targets into training validation subsets of equal sizes. And for each subset, we generated 50,000 random target source combinations. For each of the generated target source pair combinations, we trained transfer learning models, calculated the metric correlation coefficients, and applied our custom significance tests. In sequence, we calculated the transferability metrics for each of the transfer results generated in the previous steps. And using these transferability metrics as features and the transfer outcome as the variable of interest, we trained a balanced random forest classifier model using the train subset and evaluated its performance on the validation subset. To simulate the actual application of the transferability prediction model, we randomly selected 100 targets from the validation subset and calculated the transferability metrics between the selected targets and the KBL database. After that, we predicted the outcome of the transfers using our transferability prediction model and applied transfer learning to the transfers predicted positive. If a particular transfer had more if a particular target had more than one successful transfer, we also used multi-domain transfer learning using the identified sources. Based on the results of the random transfers, we have observed that transfers are very rare, with a probability of occurrence of 0.12%. In contrast to that, our transferability prediction model achieved a precision of 17%, which represents a considerable 127 times increase in performance when compared to random chance. Also, our transferability model was capable of identifying the majority of the successful transfers, achieving a sensitivity of 92%. Out of the 100 targets selected in the applicability subset, 17 had at least one significant transfer identified. Also, we have observed that cell lines are highly transferable amongst each other, with cases of cell lines achieving more than 300 successful transfers. So here we have the MCC increase distributions for the single domain transfers and the multi-domain transfers. The average MCC increase of the best single domain transfers was 0.19, and the average MCC increase of the multi-domain transfers was 0.44. So in conclusion, successful transfers are very rare, as demonstrated by the extremely small proportion of positive transfers observed in the random transfers. Transfer learning has limited applicability even when source data sets are available, as demonstrated by the small proportion of targets where at least one successful transfer was found. Finding a successful transfer in a naive manner is much less efficient than using a transferability prediction model, as demonstrated by the 127 times increase in transferability prediction performance when using our transferability prediction model. And finally, transfer learning can provide considerable performance increases on Qsar models, as demonstrated by the high average MCC increase of the transfer models. That concludes my presentation. Thank you, everyone, for your time.
Thank you, Luis. We have a time for questions, and we have one. Dmitry Shulga, what exactly is being transferred? Model coefficients?
Not exactly. We used random forest classifiers or baseline models. The transfer algorithm is applied on top of the random forest classifier model. is actually model agnostic. So what is happening is that we are using the data directly to train the models. So it's not the coefficients that are being transferred, but the data itself is being used to transfer that information using the transfer learning algorithm.
I have also a question. Is it mean that it's similar to multitask Qsar models, something like that?
Yeah, it's very similar. Transfer learning and multitasking it's very similar. There's just techniques are very correlated between the two, but the biggest difference between the two is that when you're doing multitask, usually we are trying to model all endpoints have the same importance. When you're doing transfer learning, we don't really care about the other endpoints. We're just using them to increase the performance of the particular endpoint that we are interested.
Thank you. We have also another question from Evgeny Radchenko. What parameters descriptors are used to predict transferability?
Okay, so we used 6 metrics. The first metric is the number of overlapping molecules between the data sets. So molecules that are present on both source and the target data set. The second one is based on Manhattan distance of the descriptors. So it is the average closest molecular similarity between the data sets. So it's kind of a metric that quantifies the average similarity between the two data sets. The third one is a permutation test that uses the source model predictions. So it's a test that asserts the statistical significance of the predictions of the source model. So if the source model has good predictions, we can be confident that there is a relationship. The fourth one is a domain classifier that tries to separate the target and source data sets. And the final 2 other metrics are related to the biological assay. So if it's pretty much just sees if the biological assay are the same and if the units are the same. Those are the six metrics that we use to predict the transferability.
Thank you. We have another question from Roman Yefremov. What are the exact criteria for successful transferability?
A transferability, it is a performance increase for the transfer model with regards to the baseline that is statistically significant. So if the transfer model is superior to the baseline and this superiority in performance is statistically significant, we consider that to be a successful transfer.